Hello, welcome to another screencast where I'll be discussing the history of the DNA as the hereditary material. And this is the third in a series. And you should be able to describe and explain the experimental evidence that supports the discovery of DNA as the molecule of heredity. This time we're going to be looking at experiments done by Hershey and Chase here that follow the experiments of Griffith and Avery McCarty-McLeod. So if you haven't looked at those other two, please look at those two first. That'll give this a little bit more context here. So now we are in the 1950s. The previous experiments in the earlier part of the 1900s, 1920s for Griffith, 1940s for Avery McCarty-McLeod. Hershey and Chase here seen outside their laboratories. And they are a little bit of background here about their model organism or their model system. Right? So their model system is different than the ones used by Griffith and Avery McCarty-McLeod. They're going to be using a virus here, and this is the cartoon version of the virus, and here's the virus shown in an electron micrograph. And then this virus is called a phage. And the virus that infects a bacterium in this particular class are known as bacteriophages, right? And so the bacteriophage T4, they have T2s, T6s, et cetera. So just different strains of them. But what you need to know is that they're using viruses, which are not cells, to infect bacteria. So the living organism, the living system that they're using is a bacterium, and this is what we see here. Right? So here's a bacterium, and the very famous bacterium, E. coli. These bacteriophages will infect E. coli, and you can see here all these bacteriophages on the surface of this E. coli cell here. Now, interesting thing about viruses is that they are attempting to use to discover, in these experiments, they're looking at proteins or DNA as the transforming agents, and just if DNA is indeed that transforming agent, and is the hereditary material, then any experiment that we do, we should be able to predict what's going to happen. Viruses are attractive for that because they have DNA inside of them right there, but then the rest of this head, this tail sheath, this tail fiber, this is all protein. Right? So a virus is two things. It's a protein coat surrounding some sort of nucleic acid, either DNA or RNA. Right? So these viruses here are attractive for Hershey and Chase to use in the 1950s. Now, just a little bit of other background. Let's go back over to here. We know, and they know at this point here, that DNA in the 1950s contains these nucleotides, and a key component to these nucleotides is phosphorus, right, held in these phosphate groups. So DNA has phosphorus in it. Proteins, on the other hand, so here are the proteins. These are the amino acids down here, rather. So these are the 20 different amino acids. And all these amino acids have nitrogen and carbon in them, but then their side chains, some of them have sulfur, and this is way too small for you to see here, right? But for example, this cysteine one right here has sulfur in its side chain. So the fact that proteins have sulfur, right, but they do not have phosphorus is a key thing here, and that DNA has phosphorus but does not have any sulfur in it. So these two facts right here, right, 1 and 2, are at the basis for Hershey and Chase's experiments using bacteriophages and E. coli as their model system. So here's what they do. They first take a batch of phages grown with radioactive sulfur. Now, this radioactive sulfur is just a way to tag or to follow the molecules, to follow the sulfur. And again, remember that sulfur is in proteins and is not in DNA. So they grow these phages with this radioactive sulfur. It's not, there's not radioactivity like, Teenage Mutant Ninja Turtles turning them into, pizza-eating, crime-fighting turtles or anything like that. This radioactivity is just a way to follow and to tag the sulfur. So they are hypothesizing, right? If the protein is the hereditary material, then when these phages that are grown with this radioactive sulfur, the sulfur is being incorporated incorporated into the protein coats, that is the capsids. It's being incorporated into the protein coats of the capsids of the viruses. If the proteins are the things that are infecting the cells and transforming the cells, then you should be able to follow that and track that. So they grow these phages here, and you see the phages with the radioactive protein in the capsid, but not in the DNA. Right? They then let these phages infect the bacterium, right? And I believe they're using E. coli as their organism. They then, in classic, this is the classic blender experiment, they actually put the whole thing from this flask into a blender, and then they blend it up, and when they blend it, this knocks the capsids off the E. coli, right? So they grow up the viruses with these E. coli. They then they mix it all up here in the blender. They then centrifuge that. So they take this concoction, put it into a centrifuge. And when they centrifuge it, the heavier things go to the bottom and what is known as the pellet. And then the lighter things go to the top. And the liquid on the top is referred to as the supernatant. This is the liquid on the top. And since the cells are heavier, more dense than the protein coats here, the pellet ends up being cells, and the supernatant then ends up being the capsids. And this is just evidence to then show where did the radioactivity end up. Did the radioactivity end up inside the cells? Is that what was transforming the cells, or was it the protein? Did the protein end up inside the cells, or did it stay out? Right? And so these results right here, the radioactivity in the liquid, not down here in the pellet, lets them conclude that the proteins do not enter the cells, number one. And number two, that the radioactivity, the sulfur, then is not passed on. Right? So when they take these cells and grow them on a petri dish, you get reproduction of the viral genetic material, right, that was not radioactive. Right? So no radioactive cells, no radioactive, not offspring. What do I want to say here? No radioactive cultures after that. So that was not being passed on. The protein was not being passed on from one cell to the next. So they do the opposite experiment as well. In batch 2, they grow phages with radioactive phosphorus. And again, this is a way to tag the phosphorus. And so this phosphorus ends up being incorporated into the DNA of the phages. Pardon me. They then incubate the cells with the E. coli cells with the radioactive DNA incorporated T4 bacteriophages. The phages then inject that DNA into there. They blend this up to knock the protein capsids off here. They then take that mixture and centrifuge it. And as they were expecting, the supernatant was not radioactive, but then the pellet was radioactive. And so that tells you that the DNA ended up inside of the cells. This is how they know that the DNA went in and not the protein. And then subsequently, they took those cells and were able to subculture them and get from them more radioactive viruses. And so now this is of note because in order to get more viruses use the host cells to reproduce the DNA. Right? So you get viral offspring, in quotes, that have radioactive, a radioactive nature to them. They have radioactive DNA. Then what they also did, not shown on this image here, is that they were able to take these viruses right here. Right? Well, you can tell me. Right? What would you predict would happen if they took these viruses and grew them with more E. coli? Right? Put these viruses into E. coli, then what would you expect to see here? And they continue to do this subsequent sort of subculturing, and what would you expect to see here? Right? So this series of experiments by Hershey and Chase, and here's the whole thing all together, right? So this series of experiments by Hershey and Chase confirm that DNA is the genetic material, that DNA is what's being passed on from one cell to the next, and not protein. Right? So again, you should be able to describe the background for this experimental setup, be able to explain the steps of this experimental setup. Why did they do batch 1? What was batch 1 confirming for them, and what was batch 2 confirming for them, and why and how? Okay. So again, more experimental evidence here supporting DNA as the hereditary material. And we've looked at Griffith, we've looked at Avery, McCarty, and McLeod, and in this one, of course, we looked at the details of Hershey and Chase. Thanks a lot for listening. Bring your questions to class, and we'll see you there.